Okay, welcome everyone to Toronto for this 10th Hepatitis B Cure workshop that we've been doing. So we're proud to have been doing this for 10 years now, and I'm also proud to have an extremely strong faculty. A few more people will come in. There are many people joining remotely, and we have an also very strong lineup, which we're very proud to have here, and several of you will leave later today to Washington, and I'm one of them, and that's the reason that I swapped with Adam, who was actually supposed to stand here to do the opening speech, but he will close the meeting because I might have to leave a little bit earlier, not having an American passport but European passport, trying to get through customs in Canada. It takes a while. I have experience with that, so I might leave a little bit earlier, and I apologize for that. There's a few slides that were prepared. There's 5 sessions in total, which is really a mixture of clinical and translational work and maybe a touch of basic science as well. But it's really a meeting that's also the beauty of this meeting, that has the expertise from very different angles, and hopefully we can also create a lot of discussion between these different angles, that clinicians, immunologists, virologists talk on how to get hepatitis B cured. It hasn't been an easy road yet. People working in hepatitis C 10 years ago told me, well, we'll crack this nut within 4 or 5 years. Well, here we're 10 years later and we still haven't done it. So it is a challenge, but we've made a lot of progress over the time, particularly with different types of combination treatments. And a lot of interesting data will come out at the coming AASLD, and Adam has tried to make an overview of that, working almost through the night, looking at all the abstracts, and I think I hope you will enjoy that in the afternoon. So, as I said, there's 5 different sessions. Questions can be asked here, obviously, within the room, and those who are joining remotely, there's on your screen a button to get the Q&A here to the desk, I think, right, Ricky? So then the chairs can read out that question and we can have discussions on it. These are our sponsors, and we're really very happy that all of the companies were really kind enough to support this meeting, which is, I think, important, and in a way also different at what happens at AASLD, where there's big rooms. Here it's a rather small room, but with an enormous amount of expertise in this particular field, and I hope, as I said, that we can have vivid discussions. These are the other supporters, and I'm very happy that the Toronto Centre for Liver Disease, currently chaired by Jordan, is also endorsing and supporting it. So, there is a survey afterwards, and then if you would like to have a certificate of attendance, you only get that once you've filled out the survey, right? So this is the way it's handled, and I hope that people would like to do that. And with that, I would like to invite the first 2 chairs, Marion Peters from the United States and Australia as well, obviously, and Scott Fung, who is born and raised in Toronto, and the 2 of them will together chair the first session. Thank you very much for coming, and I hope you will have a very good meeting.
Thanks very much, Harry, and we also want to welcome the attendees. Please put your mobile phones on silent. And Scott's going to introduce the first speaker.
Yes, good morning, ladies and gentlemen. Our first speaker of this session is Professor Mark Sulkowski, who really needs no introduction. He's a professor of medicine from Johns Hopkins University in the United States. And we will all know that Professor Sulkowski is a key opinion leader in hepatitis B and hepatitis C clinical trials with and without HIV co-infection. He published many of the seminal articles on the treatment of hepatitis C, and he has over 300 peer-reviewed articles in very high-impact journals. With that, Mark, welcome.
Great, thank you very much. It is a true pleasure to be here to talk to this group about combination therapy for hepatitis B. I want to point out one thing on my introductory slide is that I'm perhaps one of the few fellows of both the AASLD and Infectious Disease Society of America. And what I would point out is that does make a difference in how we think about the approach to viral hepatitis. It did in hepatitis C, and I believe it will with hepatitis B as well. So what I'd like to do, I realized actually last night that I had 17 minutes to cover this topic. I had to cut my 25-minute talk down a bit, but I'd like to go through just some general concepts. My intent is not to cover every drug in development, but to think about some issues related to sequencing of therapy and to think about some of the areas that we need to address in terms of combination regimens. I also wanted to start by thinking about just where we have been. We actually haven't had a new therapeutic entity for hepatitis B since 1998, and that was the polymerase inhibitor lamivudine, which of course is a long period of time without antiviral therapy, and we haven't made advances. So I think in general we have a long way to go, but the momentum we've had in the last 5 years with respect to therapeutics is really impressive. I also want to touch on the issue of that treatment is recommended for only people who have active hepatitis. But as we think about how some modalities may work, particularly antivirals, they may actually work better in younger people with high levels of viremia, primarily with S antigen derived from cccDNA. And we'll touch on that. It's also a population where placebo may be possible. So we'll talk through some of those issues as we get into some of the regimens. Now, what I did is try to base this on the FDA guidance that was finalized in April 2022. They essentially say that current therapies are safe, well tolerated. A key element as we think about things that we might try to do to kick the immune system into gear. They also talk about the idea that sustained viral suppression is linked to better liver histology and better outcomes, including decreased risk liver failure and cancer. Also important. And here they outline that S antigen loss is the best predictor of clinical outcomes, really highlighting the emphasis on this particular parameter. I'll come back to that point when we talk about some of their other characteristics. The other thing I'll mention, I won't go through the definition of functional cure. This group is certainly well versed in this. I wanted to think a bit more about this idea of persistent hepatitis B S antigen, so that continues to be found in the blood, presumably from integrated hepatitis B DNA, but no DNA is detected in the blood. So this is another alternative definition of cure. And the question I want to think about is whether or not this is sufficient to change the outcome of patients. Will there be an ongoing risk of clonal expansion of hepatocytes and malignant transformation in the future if we don't address this issue of integrated DNA? And in fact, in the functional recovery we see spontaneously, clearly there's not transcription of integrated DNA. So this is the FDA guidance on chronic suppressive therapy. Quite a disappointment to many, given that the addition of a CAM to entecavir in treatment-naive e-antigen-positive patients led to a more profound and rapid suppression of HBV DNA. Yet this guidance says that for a superiority trial, it is not clear what the appropriate endpoint is, and it's unclear that more rapid or more potent or deeper suppression leads to clinical benefit. Now, I'll push back a bit because we have plenty of studies that show replication, which clearly occurs on nukes, is bad in terms of prognosis. Yet the endpoint of HCC or clinical outcomes would take decades. They also then say that S antigen clearance may be considered. Sounds a lot like finite therapy, essentially making this a difficult pathway. I wanted to come back to this point, a paper recently published by my colleagues at Hopkins, Ashwin Balagopal and Chloe Teo, And what they did is looked at liver biopsies obtained from people with HIV and hepatitis B. They had stable HIV. We can talk about whether or not there was immune recovery. And they looked to see what the source of the S antigen was based on single cell hepatocyte work with microdissection and primers that they developed. What they essentially noted was that if the primary source, which is depicted by this line on the middle figure, if it's below that line, the hollow circles, the primary source is cccDNA. Those patients actually had a S antigen decline when treated with nukes for a period of time. If the source was more likely or greater predominantly from integrated DNA, the S antigen didn't budge, as you might expect. The other thing that I think is important is that long nuke therapy did lead to a decrease in the amount of transcriptional silencing. So you saw that more hepatocytes were cleared. So in the liver, less hepatocytes were infected, going from 99% in EH-positive patients to a much lower number. And we saw transcriptionally silencing with nukes. Now, if nukes are a lousy antiviral therapy in terms of full suppression, perhaps we can do better. So this is the guidance on finite therapy. A couple of key points. They do offer this S antigen positive, DNA negative option. I already raised some concerns I have about that endpoint. I won't go into it. But they endorse S antigen loss 6 months after and stopping all therapy. I'm going to come back to that as well. And you can see that we don't achieve this very often, even when we deploy interferon and tenofovir. So what about cure with direct-acting antivirals? So if we think about where the S antigen is coming from, these approaches are more likely to be effective in people who have primarily cccDNA-derived S antigen, so younger E antigen positive. Now, I recognize that's not absolute, but in general, those people have less integration. I've listed some of the drugs. I won't get into them all. For example, I won't touch on gene editing, but certainly the promise of gene editing for hepatitis B is great. The risk and hurdles that we need to cover to get there are also great. I think the good news for viral hepatitis B is that other disease states, which are imminently more life-threatening, are pushing the boundaries forward with gene editing. And I anticipate we'll be able to borrow that technology and those safety advances and adapt them to this viral infection. Now, the restoration of immune response is also important, because I want to get to the point, is there a population where we don't need to kick the immune system and increase the potential risk of therapy? Might there be a group that could be cured with antivirals? And if so, how long would that actually take? I didn't stress this, but with the nukes, they've done some modeling with a group in Los Alamos, about 130 years to eliminate hepatitis B from hepatocytes in the liver. Obviously, that's not a time span that works for drug development or for the patient's life expectancy. So I'm going to touch on a couple of things that I think are worth highlighting. The first is bulevirtide, which we know is safe and tolerable. And in the presentation by Heiner Wiedemeyer at EASL in London just this past summer, there was the notation that about 40% of patients with this, obviously, co-infection with D were not on nukes. And we did see a modest decline at the higher dose in HBV DNA. So this idea that entry inhibition didn't lower the S antigen in these patients ignores the idea that it may very well be a potent antiviral or at least contribute to reinfection of hepatocytes. Now, I'll also touch on capsid assembly modulators. I touched on the one that was tested with VBR, tested with entecavir. But these are potent antivirals, and this is, for demonstration purposes, the Alagos antiviral. And we talk a lot about these two mechanisms. I think what's interesting and what's worth thinking about is we talk a lot about this secondary mechanism of blocking the replenishment of cccDNA. Is that really necessary, or will that really occur over time? I think it is certainly important. But what we also want to do is accelerate the clearance of hepatocytes. We want to cure hepatocytes. And if we can do that with a nuke, can we do that more frequently with better inhibition of the viral life cycle over time, kicking into this secondary pathway as well? Now, I'll touch briefly on translation inhibitors, and again, I won't touch on all of these. I really don't have a lot of time left on my timer to go through this. But I think there's some important points. This is the Arbutus 729 siRNA, and what we see here is two issues. One, I want to highlight that this study that was presented demonstrates an increase in HPV-specific T-cell activation and proliferation, and a decrease in immune exhaustion, at least in CD8 cells. But I also want to highlight that in a handful of patients, well, two handfuls to be precise, there was patients who stopped both the siRNA and a nuke. And what's going to be reported in the coming week is that these individuals all still have a DNA level less than 1,000, and none have restarted a nuke for their hepatitis B, suggesting durable changes in that life cycle of the virus. I would love to look in the liver and understand what actually happened. Did we cure hepatocytes? Where is the DNA coming from? There's obviously still active replication. Now, how will we combine this? Well, there's now been a couple of studies, and I'm highlighting at least the press release for the VBR 729 plus nuke study, which in 65 patients did not demonstrate an additional benefit of a capsid with respect to S antigen decline, as well as the study REEF-1 that you're all quite familiar with, demonstrated that adding a capsid to an siRNA was not particularly effective. I want to think for a minute why that might be. And one thing is that the capsid inhibitors block encapsulation of HBV RNA. There was an hypothesis that meant you were depleting the mini chromosome, but in reality, what's probably happening is it's backing up in the cell, and you've got a lot of RNA around that wouldn't ordinarily be there that's eventually being cleared. Might that actually interfere with the mechanism of action of siRNAs that are targeting RNA? Now, that is purely speculative, but there may be an issue with respect to sequencing. I'll touch just a bit on the antisense oligonucleotide targeting translation. Certainly a very important intervention. And I think one of the most intriguing ideas is that it's this combination of the DNA-RNA hybrid complex that leads to this activation of pathogen recognition receptors, in particular in the studies presented at EASL TLR8. Now, this highlights the potential both to block viral production of proteins as well as stimulate the immune system in a way that's perhaps getting an antiviral mixed with a immunologic. And then the final antiviral I want to touch on is the monoclonal antibody. And there's an interesting poster presentation that will occur. This just shows the S antigen decline. But one of the potential mechanisms of action depicted in the figure to the left is entry inhibition. And in poster 1187 that will be presented, there's a discussion about a reduction in HBV DNA, again highlighting that entry inhibition, if that's how that's happening, and I don't know that to be true, might be an important factor to consider, particularly in a replenishment of infected hepatocytes. So I'll move to get through the immune system relatively quickly. And here, I think that when we talk about ways to stimulate the immune system, and in the HIV world, they used to talk about a kick and kill approach, which essentially involves viral latency. And it's not quite that same approach here. We're not trying to resolve latency, but we are trying to kick the immune system. And there's a couple things that I think are important to consider. One is this mechanism is absolutely critical with respect to integrated DNA. I don't see how we would use antivirals to remove integrated DNA infected hepatocytes. Now, there are a lot of different strategies to pursue this, and I won't touch on those, but just a couple. I do want to touch on the recombinant interferon redux. What you see to the right is a 1976 New England Journal of Medicine paper with human leukocyte interferon looking at Dane particle-associated polymerase activity, S antigen levels, and core antigen. This was 3 patients. So this is but in a sense, that figure looks a lot like some of those we're seeing today at upcoming meetings, obviously different markers. Interferon is a unique and interesting drug given its potential mechanism of action that I won't get into, both with respect to antiviral effect, its potential effect on cccDNA, and as well on immune-mediated response. So interferon, alive and well, approaching 50 years of human trials for hepatitis B. Now, I wanted to combine the idea of therapeutic vaccine and anti-PD-1, anti-PD-L1 into a single slide. So I selected the study that you see here with the chimpanzee adenoviral vector, which encodes inactivated Pol C and the entire S region, and the sequencing of therapeutic vaccine plus low-dose nivolumab. Now, I'll comment that I'm not entirely sure the notion of low-dose checkpoint inhibition for viral infections is valid. When we tested nivolumab, back before it was nivolumab for hepatitis C, we did see a patient cured and another with a large response, but it was at the highest dose we tested in a single dose. And this idea that infections have a different threshold than in cancer, I'm not so sure that makes biologic sense. So we have to think about what low dose really means. So to summarize a couple of thoughts on a few slides, combination antiviral therapy targeting really the elimination of transcriptionally active cccDNA. What's the right population? Treatment naive, e antigen positive, and maybe we can avoid the risk of a kick modulation and allow the so-called restoration of the immune system, or perhaps with some drugs like Bepi, this innate response. Now, what about sequencing? Well, I think the CAM plus siRNA studies tell us that we probably don't need to do that experiment again. It doesn't appear to work. But what if we knock the proteins down? And I'll take that 729 study where you've got people off therapy with still some viral replication. What if we then maximized antivirals after we knock down S antigen with RNAi, and then we treat it for a period of time to see if we could eliminate the transcriptionally active cccDNA? What's the half-life? How long does that take? I have no idea. But I know that we're not potently suppressing viral replication with nukes, and probably not nukes plus VBR. It probably needs a more potent drug, some of which are coming, and perhaps entry inhibition. I don't want to forget that as we focus on Delta. So what about combination therapy to eliminate both integrated and active cccDNA? Here, we've got a population of patients. We know who they are, and there's many of those patients. And what if in this case, we looked at a sequence where we did sort of the same thing. We knocked down antigen production, but then used immune-targeted therapy in conjunction with antivirals. Now, these experiments are being done. I bold interferon because as much as we hate it, it is a drug we know. And keep in mind that the longer you take it, the worse it is. And if we could limit the course of therapy to, say, 3 months or even 6 months, it is far more tolerable for human beings to take interferon. I also don't think we can cut the dose down. I think it has to be a standard dose. A couple things I didn't touch on, like CAR T cells or cellular therapy, very intriguing. And I will say there is a push, fascinating studies on CAR T for lupus and other infections. And the last point I want to make about combination therapies is ethical consideration for research. This was a paper published by Jeremy Sugarman at Johns Hopkins, who does a lot of work on HIV cure. They have a lot of thinking about community engagement. Now, I went through his table here where he talks about that there is a risk with stopping therapy. And he said not applicable. Well, we know that's applicable. When you stop therapy, sometimes people flare. So that's an important consideration. I crossed that out and said to comment. I've got to talk to Jeremy when I get back to Hopkins about this. And then the other one about stigma. I think stigma is incredibly common and absolutely impacts the presentation of patients. And then the last one is a need to engage patient community in cure research, particularly as we think about things like checkpoint inhibitors. So to summarize, I think that there is growing evidence that antiviral therapy can target transcriptionally active cccDNA. Will more potent replication help? How much integrated DNA is too much? And what if we could quantify that with a blood test and say, you know what, this patient has a lot of integrated DNA-derived S antigen. We need to think about this therapy, precision medicine. Immunomodulator therapies will likely accelerate cure for all patients. So I'm not distinguishing these, that you wouldn't use these, but I think it will be particularly essential if we're trying to eliminate this integrated. The drugs and the strategy sequence will matter. I don't have the right answer. And finally, I think we do need more community engagement to calibrate the risk-reward for some clinical trials and some drugs we may approach with. So I'll stop there. Thank you for your attention.
Thank you very much, Mark. Please take a seat. And now we're going to turn to Jordan Feld, who's going to talk about his therapy-induced surface decline the same as naturally low surface. Jordan is from the University of Toronto and is a hepatologist as opposed to Mark, so you're getting the alternate view. And he is really an expert in clinical research and hepatitis B. Welcome, Jordan.
Thanks, Marion, and thanks to the organizers for the invitation. A bit of a challenging topic. Is therapy-induced surface antigen decline the same as naturally low surface antigen? I will say I've spent a lot of time thinking about this, trying to put together this talk, and I'm not sure I have the answer, but I'll at least go through some ideas to think about. So here are my disclosures. And so the quick answer to this is, despite our hopes, probably not is the quick answer, that it's probably not the same, but it might depend on how you get there, and certainly I think the crux of this is that different mechanisms of action are likely relevant. And an important question is whether that's actually the goal. Are we trying to exactly mimic what we see in naturally low surface antigen levels? Maybe, but not certain that that's the answer. So I think thinking through this, some questions that need to be considered is how do we get to low surface antigen levels? So what is the mechanism of action that you're using, and is that stable once you're either on the drug and or off the drug? What are the consequences of that? And then for each of these considerations, how do they differ between the natural scenario and drug-induced declines? So let's sort of think about what happens when you get low surface antigen levels, either complete surface antigen loss or decline in natural infection. And if you sort of look at an HBV-infected hepatocyte, a lot of the clearance is going to happen by clearance of infected cells. So you can clear the virus from the infected cell with non-cytolytic clearance. You can have direct cytopathic clearance, killing of cells, and you can also just have silencing, which leads to cell turnover. So all of that takes away that infected hepatocyte effectively. Now, the alternative is this idea of transcriptional silencing. Here, the mechanisms are a little bit less clear. So here, the important thing is that at least in the natural scenario, this is not only surface antigen that is being silenced, but it's the other transcripts as well, and that may be relevant. Theoretically, you could have translational silencing. I think that's a little bit less likely, but importantly, this almost certainly happens, because if it all was the first case scenario, we would not expect to see hepatitis B reactivation with a potent immune suppression, because we shouldn't have any either HBV or infected cells. And the fact that we do see this even years after spontaneous clearance or very low S levels for a long time tells us that we do have transcriptional silencing. And I think this should be really, we need more understanding of how exactly this happens. And I'll be interested to hear the immunologists weigh in on this, because this part of immune control is a little bit less well defined. But if you think about what this looks like, so you could have it downstream where you have transcriptional silencing, or at least in theory, translational silencing, or more likely it's upstream where you have transcriptional silencing of cccDNA and probably integrated DNA as well if you're getting actual S loss. So now what happens on the therapeutic side? Now I've put the different targets in the life cycle in different colors here. In the green, I've made the case that these are the ones that I think are more likely to mimic the natural situation. So targeting something very upstream like gene editing, destruction or inactivation of cccDNA, and you might be surprised to see the direct antivirals targeting packaging or even a nuke also with a similar approach, and similarly with blocking entry, whereas transcriptional targeting late stage effects like export or immune mechanisms are more likely a distinct pathway. And so what's the case for that? Well, the argument is that, as I made the point, with the naturally low surface antigen levels, this is through loss of infected cells, transcriptional silencing, and that you have all the transcripts are low. So what happens when you do this just with a nuke? So let's just sort of look at nukes as a simple DAA strategy. And we know that with spontaneous surface antigen loss, it's fairly similar to nuke-associated antigen loss. So this is a big meta-analysis looking at least the rate of loss, and you can see that the pooled annual rate of surface antigen loss is about 1% per year, and interestingly similar between patients that received long-term nuke therapy or were not treated. So that's similar. You see if they get loss with a nuke or you get loss with natural clearance, durable in similar proportions. So you can see that the probability of staying S loss, S negative, with either interferon or a nuke on the left, or with interferon, spontaneous clearance, or a nuke is very similar in either case. And finally, what it looks like is that it is the level of surface antigen which sort of drives this. So if you look at other factors before someone goes on a nuke, you can see whether they're e-antigen positive or negative, with the genotype or the HBV DNA, small numbers, but don't have a big driving effect here, but really it's about the baseline surface antigen level and then how quickly it falls. You can see in the bottom curve that the slope is greater in those who clear. So what can we learn from the nukes? Well, the surface antigen decline is generally slow. It's roughly the same rate as we see in natural infection. You get clearance at about 1% per year. If that happens, it's durable, and the pattern is fairly similar, that you get sort of a petering out of surface antigen rather than a flare followed by clearance like we see with E seroconversion. I think that's an important distinction, that those are really different. So it suggests that maybe the mechanism of surface antigen loss with a nuke is similar to what we see in natural infection. So this is really being driven by clearance of infected cells, driven by either cell turnover and prevention of infection of new infected cells, and that really immune clearance may actually play a very similar role in these two scenarios. So if you kind of think about it, a nuke or any other direct antiviral really sort of allows people with active disease to, in quote, survive or tolerate their infection until spontaneous surface antigen clearance occurs, because it's not really accelerating it that much as allowing patients to get to that point. So if you sort of think about any direct antiviral, you can sort of think about putting in a nuke here, and all that's doing is blocking DNA replication, but what it is doing is it's preventing the infection of new cells. So turnover of infected cells continues to happen with minimal infection, and eventually that leads to low surface antigen levels or loss. So the important point about this is it may be slow, but when you do this with a DAA, it might be much more similar to naturally low surface antigen levels and clearance. And I think that's worth considering, because I think there's been a little bit of less enthusiasm about using direct antivirals because of the lack of S loss with CAMs, but maybe I think thinking about this may actually be more similar to the natural surface antigen loss or low surface antigen levels is worth consideration. So, but the bigger question is, does the actual level matter? And a lot has been made of this idea that if we, from these data showing that if you can get someone below a surface antigen level of the magic number is usually 100, we know that's associated with a fairly high spontaneous clearance rate after stopping at least nukes. So then you see a lot of trials, and this is just one example, reporting that most patients start above 100, and then with an antigen-lowering therapy, you can get them below 100. So the real question is, does that actually matter? What does that actually mean when you do it with a therapy? So I think it's also important to consider that how you get there differs between the different approaches to bringing down S levels. So if you think about translational control, so the transcript targeting approaches, siRNA or antisense, you can do this later in the life cycle with subviral particles, things like NAPs, or even post-secretion with monoclonal antibodies, and all of these are going to bring down antigen levels, but they're going to do it at a different time in the viral life cycle and may have different effects, because they may have the same impact on antigen levels, but whether that will lead to the same impact on immune recovery and or other effects is less clear. So it's also important to consider whether what you're actually targeting is the same. So does it matter whether you're targeting the same surface antigen species, the small, medium, or large surface antigen? And some data suggesting that there may be differences between these. I think it's still a bit unclear, but this might be a key to distinguishing between integrated and cccDNA-derived surface antigen. Of course, whether this is upstream or downstream is potentially important. And then I also think this idea of whether other transcripts need to be low or not is important. When we see this with naturally low S levels, we see naturally low everything else as well. That's not always true when we start bringing in these new agents. And interestingly, now we see patients getting ASOs and siRNAs where the surface antigen level can be lower, sometimes even undetectable, and they're still e-antigen positive. And that's kind of strange. That's not the way we usually think about hepatitis B. And I think it's important that we understand, if we're comparing these paradigms, that they're not really the same, because when you do this in the natural history, all of those transcripts are low, meaning that there really is some probably immune guided transcriptional control. And then, of course, there's this question of the chicken or the egg here, so that we know that the idea here, the idea of bringing down S levels, is that you're going to bring in immune recovery. But of course, it could be the other way around, that it's the immune recovery that's actually leading to the low S levels. And this is a continuing, ongoing discussion, which doesn't have a clear answer. But there are emerging data, and Adam and I discuss this all the time, that there's not really great data that low S antigen levels actually really reinvigorate the immune system. But there are slowly coming data to suggest that this might be true. This is just one paper from a couple of years ago that showed that you do see that patients who have, this is not with treatment, but lower S antigen levels below 500 or high S antigen levels above 5,000, the patients that had lower S levels were able to have a better response to HPV stimulation. Their HPV-specific T cells produced, in this case, just one figure, but were able to produce dual cytokines compared better than the high antigen levels. So suggesting that that lowering S levels may still be helpful in terms of immune recovery. And maybe even if we don't get loss, really the idea of this is lowering levels will reinvigorate the immune response, and you just need to do it a little bit to sort of get you over the finish line. That's sort of the hope with this, but I guess the question is how low do you need to go? I think it's important to consider that it might not be right. And this is just an example which I'm not sure applies to actual patients, but it's a nice study that Adrian Cooper did, a PhD student with Adam, showing that if you think about the combinations, you've got to be careful about how you potentially sequence them and combine them. And what he showed here is on the left that if you give an siRNA, this is in cell culture, but you see that you get a reduction in antigen levels and replication, and you get a diminished core antigen specific T cell responses. Now, if you give conditioned media with TLR7 or TLR8 to stimulate the response, what you see is that you enhance the core and S antigen specific responses. But if you give an siRNA with that, what you see is that you actually blunt that response in a sense that you need some antigen there to maintain the response. And obviously, whether this actually happens and whether this is truly physiologic when you replicate this experiment may be not so clear, but I think it's an important consideration when you try to think about the combinations. And then perhaps more concerningly is this question of, can we actually really hope to reinvigorate T cells and the immune response more generally? And I think a lot of data, this is just one in the Hep C field, shows that even when you take away the antigen, so we're not lowering the antigen, we're curing the patients, no trace of Hep C that we can find, and you still have this so-called immunological scar where if you stimulate those T cells, all they do is they come back to form a similar to a terminally exhausted T cell, so that they're really not true memory T cells, and that's going to be a challenge. If that's all we can do when you take away antigen, can we really hope to do that by just lowering antigen levels? And Georg's going to talk about what a functionally cured T cell looks like this afternoon, so hopefully that will shed some light on this. But I think it's also important that in this, you did see a very clear difference between spontaneous clearance and treatment-induced clearance. So maybe our goal, rather than low S level, should be really more trying to mimic acute spontaneous clearance, where we know that you have very good, robust clearance and a durable protection. And then I think this sort of question of whether our goal should really be matching spontaneous low levels is also a question mark. This is a classic study from Carlo Ferrari's group many years ago, which sort of raised the question of whether nukes could reinvigorate the immune system. That's not really the focus here, but what I would really look at here is just if you look at the CD4 and CD8 T cell responses, and you see that patients that are inactive carriers, the ones that have low surface antigen levels, the goal that we're trying to achieve with our antigen-lowering therapies, actually have pretty paltry immune responses to HPV stimulation. So I think it does raise a question that when you actually get so you can see that the nuke-treated surface antigen loss were the ones that actually had the best response, and even spontaneous S clearance had less potent responses. So I think it just raises some questions about what we're actually trying to achieve. And again, the acute clearance was the most potent response. But before we lose all hope, even if they're not the same, maybe surface antigen reduction may still be beneficial. And Mark didn't talk about this combination specifically. He talked about the REEF1 of combining an siRNA RNA in a CAM. But the REEF2, I think, is interesting. Again, no S loss in this study, but I do think it's interesting to look at what happened when these patients came off nukes. So if you look in the right panel, you see at the DNA or the ALT. In the gray is the patients who got placebo, and in the nuke is the combination patients. And although they didn't clear S, they didn't have the same rebound. So they sort of had this kind of plateauing, something changed. And I think it's a question about whether there is some stability of off-treatment response and whether that's actually leading to some degree of immune control. And as we're going to hear more about this afternoon, and Mark touched on, is with the Arbutus 729 agent, you did see this recovery of T-cell responses with reducing exhausted phenotype and increasing T-cell activation. And there's a talk on this afternoon, so I won't go into it anymore. But so finally, to summarize, therapy-induced decline is likely not all that similar to naturally low surface antigen levels. I think it's probably most similar with direct-acting antivirals, nukes, CAMs, or anything else that leads to surface antigen loss through similar mechanisms to natural clearance and primarily enables it rather than accelerates it by blocking infection of new cells. But the important point here is that it may be very slow. More rapid surface antigen decline is likely fairly different from naturally low surface antigen levels. Reassuring that there's some emerging data to suggest that immune recovery is possible, but I think we have to be careful over interpreting these data. And this may be enabled to be enhanced by immune targeting agents. I think the message for me, as I really scoured the literature to try to find data to really compare these two scenarios, is we don't really know a lot about this, so we need more reliable immune evaluation, I would argue, with every early phase trial of new agents to understand this phenomenon. And I would encourage people doing these trials that this is going to help you. It costs you up front, but it's going to help you guide which combinations, the duration of trials, trial design, and perhaps most importantly, interpretation of your results. And a lot of these sort of hand-wavy comments about ALT flares post-treatment or whatever indicating immune activation, these really aren't adequate. We really need to understand whether or not we're actually engaging the immune system and leading to immune control. And with that, I will finish and happy to take any questions. Thanks.
So we'll have a question session after all the speakers have finished. Our next speaker is Professor Massimo Levrero from Hopital de la Croix-Rousse in France, where he is Professor of Medicine at Université Claude de Lyon in Lyon, France. And as you all know, Professor Levrero is an international expert in viral hepatitis, is actively involved in several clinical trials and research in hepatitis B and C. He has authored over numerous articles in the field. Massimo?
Thank you for the nice introduction. I would like to also thank, I don't know whether Ari or who else, for the title, Preclinical Therapeutics with Novel Mechanisms of Action. Okay, it's everything and nothing. And so what will I do? Disclosures. Okay, let's start with the landscape of what is going on in terms of targets for treatment. And yes, Great Expectations is the original one, the 1946 film. We have a lot of excitation, but up to now, truly, nothing really ended up as we hoped. And let's go a little bit more deep and think about the biological correlates of cure. Essentially, we have, I will not enter into the definitions of partial, functional, complete, sterilizing cure. Mechanistically, we have three main mechanisms: silencing cccDNA, reduction of the pool of the cccDNA, elimination of infected cells. The questions are what we want to do, control or cure? What to target, what to prefer? Obviously, we go for functional cure because we are not able today to think about something more. And then, is cccDNA silencing possible? Is cccDNA elimination possible? And is silencing propedeutic to elimination or the other way around? Going from the clinical point of view, we can classify the whole thing in a different way. We have a lot of drugs that do different things. We can think of them as inhibiting replication, diminishing antigen load, or stimulate the immune system. I will not further discuss the first two categories because somehow especially the first one, because other people will do, and there is a lot of clinical work on that. So I will try to stay away from the clinical data and try to reason on other points, about other points. Just I want to say that blocking replication, cytoplasmic replication, capsid related replication is important, and it may affect indirectly cccDNA de novo synthesis, the intracellular recycling, and the important coating of relaxed circular DNA containing capsids. And this is important, and entry inhibitors and CAMs may inhibit cccDNA establishment. I will not go into that because it was already covered. For the antigen reduction, you just heard Jordan, it may restore innate and HBV-specific adaptive immune responses. We have few data that prove this, and we will hear this afternoon about the data that were just mentioned by Jordan with the Arbutus drug, that are some of the few data that we have directly from patients. So I will focus essentially on three things, and you will see how I will do it, and probably will smile later on. So targeting cccDNA, targeting HBX, and immunotherapy, immunomodulation. Why I added targeting HBX? Because X is a crucial protein in the control of cccDNA function, and this comes from a lot of studies. And if we go a little bit deeper, I will touch first the HBX inhibition with different approaches, its relation with silencing of cccDNA transcription, and later on I will go to strategies that go directly on cccDNA stability. First question I want to a little bit to address is, can cccDNA be silenced or epigenetically silenced? What we know from clinical studies is that transition from to the E-negative chronic hepatitis infection is associated with the reduced cccDNA transcription activity. And we define cccDNA transcription activity as the ratio between the number of CCC molecules and the amount of 3.5 transcripts, because this is what is really defines whether or not there is transcription from the cccDNA. And this was showed also in several other papers, including a paper that probably Barbara Testoni will mention this afternoon because it's related to biomarkers. But there are data that confirm that with E-negative status, cccDNA transcription activity goes down, but does not go completely down. And there are data that in patients with long-term NUC treatment, again, is a paper from Barbara Testoni's group, you have indeed a silencing of the cccDNA with epigenetic changes. And the two arrows there in telvibudin-treated patients is related to cccDNA chip, in which some histone marks of repression, K27 methylation, is up, and the corresponding K27 acetylation is going down. So there is some epigenetic control. And the last panel are some data, recent data that we generated from patients analyzing the liver biopsy from patients with a functional cure versus chronic hepatitis B E-negative. And as you can see, there is much less cccDNA, much less DNA in the liver. In the bulk analysis of the liver samples, you have less 3.5. But if you look at the transcriptional activity, in functional cure patients, you still have some patients that have fairly high transcriptional activity. This means very little cccDNA molecules there, but they are active. And this is something that we have to keep in mind. And so if we go to targeting HBX, because it's the crucial protein that regulates the activity of the cccDNA by inducing the degradation of the SMAC5/6 restriction factor. If you target the interaction between HBX and DDB1 degradation complex, you indeed have a less transcription, but the effect, as you can see, is not dramatic. And this was obtained with a drug that came from a screening, but it's something that the people working on hepatitis C know very well, and it did, because it was used as an antihelmintic that was used in hepatitis C before the real direct antivirals arrived. And you can also target the NAD8 as another pathway. I have no time to go into all the details. The inhibition of a NAD8 with a specific drug lowers cccDNA transcription and lead to a cccDNA destabilization. Look at the effect. Is 30%, 40% is not big. And targeting the interaction between HBX and the 20S proteasome, again, and this study was published in Hopital, and there's also a part of the work done in vivo in mice. Dicumarol and NQ01 inhibitor blocks cccDNA transcription and promotes, by promoting HBX degradation. So there are indications that you can work out a block of HBX. This is completely preclinical. We have no idea how to convert this in something that may work in patient yet. And some interesting data come from the work of Maura Dandri's group. They published this year in Gut a paper in which they showed that by using siRNAs, the same siRNAs that we use in patients that you heard about, since the target of this SI actually go to a region that induced the degradation of all transcripts, they demonstrated that in these chimeric mice, indeed you have less transcript for X protein, and you have less X protein, and by doing this, you can induce again the activation of the cccDNA. So it's the proof of a concept that a siRNA may have a second mode of action in which, besides HBV RNA degradation, you can hope that there is some cccDNA transcriptional silencing. But if it is true, why this does not happen in patients? Because finally we have no idea. Or we didn't look well, or there is something else that is not really clear for us. And here, again, I was scooped by before, because somebody already said that interferon is coming back. So I used the DeLorean 1985 car, renamed PEG interferon, and Back into the Future. Interferon is coming back. It's coming back in the delta field. NAPs work, but they work because interferon is there, I suppose, or I fear. But we know from the Stefan Urban work that interferon has a very special activity in delta infection because it's able to block NTCP-independent HDV spread. And this is why bulevirtide plus interferon works better, whether we like it or not, but it works better. If you look at the graph from the NAP studies, you have in the red rectangle a phase of the treatment in which interferon is not yet there. The good or bad ALT flares are not there. I agree with Jordan that talking about ALT flares is not biologically relevant. It's clinically perhaps important, but biologically it does not help us to understand. And in that phase, HDV RNA is going down. So perhaps NAPs have some other activity besides entry inhibition or post-entry inhibition, as was demonstrated in some models, that has nothing to do with HBS decrease and ALT flares that induce the functional cure, and maybe there is other activities directly on the replication cycle. Let's go back to the animals, to the chimeric mice that were studied, and I presented before from Maura Dandri. They showed that the PEG interferon is indeed doing the same thing that a siRNA. There is less pegylated interferon in this model, and PEG interferon is able to induce again to target SMAC56 and to induce the re-expression of and the decrease, the block of the HBV RNA production from the cccDNA. And this was just a confirmation of a lot of data that were published before. We published that before, besides the well-known activities on the stabilization of RNA-containing capsids, besides the activity that is there but is slight and not very big on cccDNA stability through deamination. there is a clear epigenetic effect on cccDNA with silencing. Whether this is in all patients, we don't know. Whether this is contributing to some of what we were seeing in patients, perhaps yes. There are a lot of other drugs that are controlling the epigenetic of the cccDNA, but forget about them, because they're not specific. There is a lot of problems, complexity of responses, functional redundancy, duration of response, off-target effects. So we will not go there for silencing. But looking really at the preclinical data, we are coming to a scenario that looks more complicated than what we were thinking before. Because in a study that was presented at the JBB meeting 2 years ago, Mauro Dandi's group showed that shutdown of HBV transcripts promotes the reappearance of SMAC5/6. I already told you that, and cccDNA silencing, that's okay, but there is no change of the post-translational modification on the cccDNA, of the histones on the cccDNA. So there is no active repression, but more something like cccDNA compaction without active repression. This was what they proposed according to what they were seeing. And there is another paper that was published last year from Zheng Yuan group in Shanghai, in which they showed that with interferon, the accessibility of cccDNA to MAC-bind nuclease is much reduced. So again, you have something that cccDNA is less accessible after pgRNA, pegylated interferon, sorry, but there is no active repression probably. And what does it mean? Let's go for a cartoon trying to explain that. On the left, you have an active cccDNA with the transcription factor, coactivators, and acetylation marks. On the left, all the possible flavors of silencing. You may have a cccDNA that is open, but is deserted because there are no transcription factor, no coactivators. So the acetylation marks can be there or not, is not important. And you have the other possibility. I didn't see before the clock. Sorry. The other possibility that you have a compacted cccDNA. What was proposed by Mauro Dandi's group with siRNA, loss of X, but no changes of the cccDNA epigenetic control. And you may have a repressed, like in the long-term NUCs in the paper from Barbara Testoni group, in which you have some post-translational modification of the instance. And all this may have a lot of impact on what we are doing, because probably we are treating as silenced things that are different, and we are trying to change and to use drugs to do things on a completely different scenario of population of people. And it's like in the clinical trials. If you put everybody in, you may lose the effect, and if you do you select too much, you only treat 1% of the population. But we have to know before to make the decision. And in HIV, as was already mentioned, people are using the so-called the shock and kill. Perhaps we have to try to do something similar in HBV, like shock and degrade. Try to open the cccDNA or make it accessible. And indeed, this has been done in the same paper I mentioned before, by Zheng Yuan in Shanghai. They used the histone deacetylase inhibitors, and they obtained some more accessibility and degradation of the cccDNA. I wanted to tell you a few things on the direct targeting of the cccDNA, but I was too long. So I just will go to one single slide. And it was already mentioned, the potential for direct targeting, genome editing of the cccDNA, a lot of limitation, a lot of strategies to change this, and possibly one new approach may be base editors as compared to the classical CRISPR-Cas9 approaches that are catalytically disabled or inactivated Cas9 that generate single-strand breaks and not double-strand breaks, coupled with the deaminase that is able to act on single-strand DNA to edit the genome at the specific target site. And at the HBV meeting a few weeks ago, the BEAM team, in collaboration with Fabian Zulim and Barbara Testoni, they showed indeed that you can inactivate cccDNA in vitro and in vivo introducing specific mutation that abrogate HBV replication and silence viral protein expression. The big point here, like for the other approaches, is what is the fate of the cccDNA. Barbara Testoni-Guada-Martinez published a paper in which they showed that the residual cccDNA that is strange, is manipulated, is edited, is still transcriptionally active. So we have really a problem there. Immunotherapy, I will not touch it because I was afraid to have I didn't dare to put Georg Lauer photo there, but I could do that with Adam, so I will not talk about that. And finally, the only slide before ending was this one, and it's already been mentioned. The epigenetic scar is very important. We have really to go into that. And there are also few evidences that something similar is also going on in hepatitis B in addition to hepatitis C. Thank you very much.
Thank you very much, Massimo. So this session is now open for questions and discussion. There are microphones around the room. Put your hand up, they'll bring you one. And for people online, please send your questions. We will see them in the iPad. Okay, and please state your name and affiliation.
Robert Gish, Hepatitis B Foundation. It's a great start to a wonderful meeting. Thank you. What is the data that supports ASOs as direct immune modulators versus both versus an antiviral agent? I think there was a recent editorial that discussed this concept.
Who are you asking?
And nobody wants to answer.
I can take a stab, but I mean, I think there's arguments for and against. I think, at least from my mind, the most convincing or the most suggestive data that is not purely an antiviral is really that the GalNAc conjugated version is less effective than the non-GalNAc. So the naked ASO that's not GalNAc conjugated goes into primarily non-parenchymal cells, and that's more active than the GalNAc conjugated one that primarily goes into hepatocytes, which would argue that I do think it's specific. So if you give this to somebody who doesn't have Hep B, you don't get any immune activation. So it is, you are getting, it is critical that you're getting the oligo binding to HBV RNA, but that is probably not being driven, at least not exclusively, by what's happening in hepatocytes. So my take on it is it's more immune modulatory. But that's, I'd be interested to hear what others say.
Well, I would just add, I think it's also very consistent with how pattern recognition receptors function, and you could envision how that might play out in vivo in a person who has these complex RNA-DNA hybrids. So I think it's consistent and fits into a story.
Microphone 2.
I have a comment and a question. So the comment is about the story of, the scar T cells, because it's come up twice now. And I think we shouldn't be surprised. We know already in HBV that lowering replication to really low levels doesn't restore immunity, because otherwise you would have much more clearance. So I think that's not the problem. And I think people see this a bit more dramatic than at least I do. And there's good data now from Rafi Ahmed's group lately in cancer, where they showed actually they see the exact same profile, the same epigenetic even, fixed state of these cells. But then when they add anti-PD-1, you get some recovery that we see in patients obviously too. But now they have a story where they show that if you add low-dose IL-2, actually don't get even more cells, you also get really different cells for the first time, because when you only give anti-PD-1, you get more of them, but they're still kind of the same. But so you can really reprogram, it seems sometimes, or at least increase new populations. And I think that's what we need to think about and what we're going to try, right, with the vaccines. I'll come later to, I think IL-2 could be from CD4 cells, so I think the CD4 T cell response might actually be really critical to this. So I think it's not really a new challenge in HBV. It kind of just confirms maybe why reducing the replication didn't do the job. And Massimo, do you want to say something right away or to this? Or
right.
And then I have a question None of you mentioned that, the loss of HBs antigen, especially maybe in the later stages, when we are, let's say, in IC patients to functional cure or in treatment, no one mentioned antibody HBs antigen complexes that might mask what we measure. And you will see, I will come back to that later in my talk. I just wonder, is it really that at the end we get rid of all the cccDNA, we get rid of kind of all the antigen, or do we just mask it and make it inactive? And is that what really switches the balance and what also makes the situation more stable? And I think we really know very little about it, it seems to me.
Yeah, it's a great point, Gabriel. I had a couple slides on that, and I took them out for time. But because it is one of the things in terms of thinking about the point that I made about transcriptional control that we don't really understand. Another alternative, of course, is that there still is continuing surface antigen production. It's just being outproduced by surface antibody. And I agree with you that understanding, and there certainly are data that I think the question of whether it's actually surface antigen loss or appearance of surface antibody is an important question, because we can only measure we're really only measuring free antibody when we don't look at immune complexes. So I definitely take your point that that's important.
I would like to come back for 1 second on the scar, or the epigenetic scar. I mean, it's obviously is not. I agree with you, but it's not something that is just always the same everywhere. Epigenetic is 50 shadows of gray in how it is imposed, and different complexes, different actors, different targets, different level of suppression, different level of how easy it is to go back to different levels of plasticity. When you methylate the DNA, you probably lock it a little bit more, but we are not talking about that in this case. We are talking about more classical histone post-translational modification profiles in specific loci. Is also something that was clear for not only for the viral replication. I mean, in cancer, in hepatitis C, you eliminate the virus, and you still have a scar on a number of genes that are related to proliferation that are there and stay. Several papers from Thomas Baumer lab and others. So I agree with you. It's not dramatic, but it's something that we have to consider, because probably we didn't go into classifying it related to different phenomena. So this is why I put the slide, probably why you also put it, because we don't understand that. And again, a similar thing for the immune complexes. We don't touch it because we don't measure it as doctors. That is the point. So we can put a slide, but we don't manage we are not able to drive that. And so we prefer not to elaborate too much. But as scientists, this means that, are we sure that when we have S loss, there is no S transcript, S protein? And you mentioned CD4 responses. Your data, there is also data from Singapore that go in this similar direction, in which some data point out that there is still transcription in the liver of patients with a functional cure, as I showed. Probably there are immunocomplexes there. It's possible. Why not? It's full of this in several other heart diseases. I agree with you. But we have not. There was one essay that was an investigation essay from Abbott that never really arrived in our hands properly. There were only a few papers.
In the complex?
Yes, and that was better than doing the immunocomplexes by ourselves homemade. But still we don't know, so we don't mention.
Complicated. Adam.
Yeah, I mean, I'll follow up on that, Massimo, because those of you that are going to leave it early, there's 2 posters at ASLD this year. One is looking at biopsy functional cure patients. 20% of functional cure patients have S antigen positive hepatocytes. And there's a study from Daryl Lau using this Abbott assay showing that their immune complexes in functional cure patients out to 24 years after functional cure. Years, right? So it's definitely there. It's just a matter of whether we can see it, right? And this comes back, and maybe I'll sort of tap Pietro for this too, because the endpoints review is coming up at ASLD, and there was discussion at the endpoints meeting in June, walking back from functional cure and thinking about partial cure, and is there really a difference between these two versus just masking the S antigen or not? And I think that is a debatable question, actually, at this point. And then I want to come back to Georg's point too, and maybe this idea of the scar, because you might know better than everybody, because I mean, we've all sort of referenced this paper as a great paper from their group, but I mean, presumably within individual clones of T cells, there's a spectrum of differentiation, right? I mean, how much do we know about the states of it? And they're not all terminally exhausted, presumably. I mean, there must be. Do we know? Do we have that resolution? I know this may be a question that you're working on. I don't have it yet, but.
So I think we kind of unfortunately do have an answer bit from Robert's group, and we generate similar data right now ourselves, which is that if you do single cell sequencing, you really see that it's really pervasive. So pretty much while after you remove antigen, you do see, a complete takeover of what we thought would be good, like stem-like cells expressing TCF1, that they all still have these same basic features, and it's really kind of 100% of the cells. So that seems, and that's the same in cancer too. You just see that there's a different state induced, and it really goes all across. But as I said, the data from the cancer, really from Rafi, really seem to indicate that there's opportunity if you do the right intervention. And also we know from natural history of HPV, we have patients 30, 40 years in, they achieve functional cure, right? So if we assume that that's the immune system, which I would have no other idea what it could be, then it seems to be somehow reversible. The question is how.
Microphone 2.
Yes, hi, Vance Vasily from GSK. Actually, my question becomes a little bit redundant after Georg, but still I want to ask it. So Jordan was kind of suggesting that in our attempts to stimulate immune system, we should aim to mimic really what happened in acute resolvers. And obviously that has two dimensions on one hand in terms of magnitude, but on the other hand in terms of signature, transcriptional activation, etc. So do you think, maybe Georg, do you think that single-cell RNA-seq is the right strategy to look towards these signatures? What is the right path one needs to take to define those and the functional level comparison?
Studying transcriptomics with single-cell RNA-seq is great. You have beautiful images, you reconstruct a lot of things, and you get a lot of information, and probably you can interpret phenomena much better than with bulk RNA-seq. And that's clear. And I will not advocate the opposite. But where I'm a little bit more skeptical is when you think to relate these findings to the virus with the same level of depth. Because we have to remember that when everything goes well, you have a couple of molecules of cccDNA in the infected cell. Sometimes you have more, but let's say that in most patients you have levels that when it's 10 is very good. And single cell technologies adapted to so little template will eventually allow to have a coverage that is enough to see the transcripts. You may do Illumina sequence and have your nice skyscraper profiles, but you will never know which transcripts are done. And if you want to go a little bit deeper and do CHIP or ATAC-seq, you're lost because you have not the coverage. Just technically, it's impossible to have enough coverage. So I think that you want to go to a single cell, you have the money, academics have not, you have the money, or not all of us, you have the money, you go for it, that's good, you get a lot of information, but then you will correlate this with bulk data in terms of, also the data that were produced in Baltimore at Johns Hopkins with the single cell and PCR, not sequencing. They are quite difficult to interpret if you really want to go to the end, because you amplify a lot, also in RNA-seq you amplify a lot. So finally, you have troubles there. Just want to comment that.
Thank you very much. There's a question for Jordan and Mark. will different combinations be required for different stages of hepatitis B?
Your topic was combinations.
Well, so I think, even listening to this discussion, I think one thing to recognize is that the hepatitis B natural history is heterogeneous. Even functional cure, you can envision that as someone with no cccDNA, they're truly eliminated, someone who has silent cccDNA, and then you can think about someone who's got modestly or low active that's masked by antibody. It's interesting, every stage is fairly heterogeneous, and our understanding and our ability to define that phenotype easily is limited. But I do think there is room to think about precision medicine. I don't think we have the tools necessary to apply that in a way we'd like to right now, but I do think there's ways to think about different approaches leaning towards DAA or antiviral approaches in people earlier in infection, and leading towards more complicated, perhaps, immune mechanisms later. Now, that's a vast oversimplification.
I'll just weigh in. I think it'll depend a lot on what ends up working. I think some combinations will probably work across the spectrum, and some combinations will be much more specific. And you can think about rationalization of one versus another, but it sort of raises a question that I was going to ask Georg just in your discussion about this sort of terminally exhausted phenotype, like whether that's true across the phases, because the data from Bertoletti and others showing that really young patients are able to mount responses and older patients are not. But then whether you call it immune tolerant or not, you have patients with super high replication that somehow are not generating a very, at least not active inflammation in the liver, which again is not so clear in understanding why that is. But it's a little bit surprising to me if you look at the T cells in those two patients and two groups, like someone who's got very good immune control but an inactive carrier versus someone who's got very high replication with no inflammation, and that they both look exactly the same and both extremely terminally exhausted. I don't know if you're going to talk about it later, but if that's not the case, then I think it's relevant.
I think we should talk about it later because there are a few questions.
We also have to let Harry ask a question.
We'll hold that
He's
for
the
1
chair
second.
of the meeting. So. But
Yes.
sorry.
Let me make maybe one comment, because I will not touch on it later, because we are still in the process of, Jordan knows we are having produced data with Adam and Andrew Boonstra and others using single-cell technologies. It actually was surprisingly not so easy to see major differences in these profiles. Now, I think the ones in the immune-tolerant phase, we will see a bit more, and most likely we would see the most in patients who actually have ongoing hepatitis, but those are the ones we have the fewest, and that's a bit our problem. And we had a bit robust discussion at the HPV meeting, because Michael Hoffman's group in Freiburg, they've been looking at this too, and what we all, I think, see and agree on is that in IC patients for sure, and in treated patients, we don't see this extreme exhausted phenotype, like super high. Actually, they have PD-1, but we don't see the downstream effects, but maybe not so surprising, because mostly it's treated patients or IC patients, they don't have high viral load. So I'm not so surprised that we don't see it there. So it's in HPV, as always, it's so much more complicated, than in HCV, and we just don't know enough yet, I think, to really
Thank you
figure
very
out
much,
what's going
Georg.
on.
So we have some questions from online. We don't want to ignore our online group. And so the first question is from Xing from FDA, who's asking Mark and Jordan, do you think patients with natural immune status, what does that role play in the level of integrated DNA? So if you have a heightened immune system, do you have lower iDNA levels, perhaps? If you could explain that.
So you mean, someone who's spontaneously cleared, whether they still have integrated DNA? Is that?
Well, Mark said if there was immune control, they had lower integrated DNA, but maybe I didn't hear that. That's what I heard, and that's clearly what
The FDA question.
the FDA heard.
Well, I'll let, now, that's not, I guess, the way I would think about it is that you want to silence transcription of both sources of DNA. And how that actually occurs, I think we've touched on that, and I'm not sure I know how it occurs, but I do think that there are likely people with spontaneous functional cure who still have integrants, but it's silence. But then again, I'm speculating.
But I think the opposite is also true, that you have people that have effectively no functional cccDNA left or very, very minimal amounts, but are still surface antigen positive, all from integrants. And those patients, we don't call them cured because they're still surface antigen positive, but they're really the same as the people who, and I actually think that's a real big issue for the field if our only endpoint is surface antigen loss, because some of these patients, their prognosis is probably the same as someone else who has no cccDNA.
Well, I think that's sort of the debate. I mean, I do think there are people where you could say the hepatocyte is cured. There's no cccDNA, but what does that mean with respect to the clonal expansion of those cells and the cancer risk? And is that really what we want, or do we really want to render people without any active transcriptional activity?
We have to remember that integration is an event that occurs when the hepatocytes is infected, not with the other phenomena like recycling and whatever. So the longer the replication goes on, the longer you are exposed to integration.
Right.
And for a longer time, you have the chance to develop clonal expansion of few of them, and so cluster of HBs positive hepatocytes.
Harry, do you have a question?
Yeah, I actually had two short questions. First of all, let me stress how much of a delight.
There are no short questions here.
Okay, well, it's a bit longer.
Prerogative.
At least no short answers.
Let me stress what a delight it is to have an open discussion with people here in the room, something which we really all have missed over the last couple of years, so I really appreciate that. I actually two questions to Jordan, but the panel can answer as well. The first thing is that you're talking about the natural history and treatment with nucleoside analogs where kind of the S antigen is fading away over time, right? It would be actually good to go back to the early interferon studies where we saw flares and then people became S antigen negative, particularly in the E-positive population. A lot of people don't read literature, which is 20 years old, but let me get to the question is what would be for you an acceptable duration of a curative treatment for hepatitis B? Because it basically says that we might need to treat longer, right, than half a year or a year.
Yeah, and I mean, I would say that if you have a well-tolerated, like if you have something that's similar to a nuke, but you had to take it for 3 years or even 5 years, but it led to S loss in everybody, we would love that. The problem is that it's just hard to design trials that way. Like that's just a practical issue of trials. But in terms of acceptability for patients, right now we're putting them on indefinite therapy. So if we could put them on indefinite, less 10 years, or to me, long therapy that is safe and well tolerated is totally fine. And the idea that we need to get something that's 48 weeks is totally artificial and probably, in my mind, at least with direct antivirals, not realistic. But I would be quite comfortable with longer therapy.
And I would just add that the idea that nukes are truly inhibiting the viral replication process is, I think that's an area where we've kind of fooled ourselves with the not detected in the blood. There's a lot going on in the liver, and I think there's a lot of potential, much room to move towards truly inhibiting the virus using entry inhibitors plus CAMs, et cetera.
But Harry, just to counter there, like I agree with you about that the fading out isn't necessarily the case with interferon. I was really making the case that it is that way with nukes.
Yeah.
But I think even with interferon, you're right that it's mostly the E-positive patients. And I guess I made that point really to stress the point that there's a lot of focus on the sort of therapeutic flares, and I think there's more and more question about whether that's really a real phenomenon.
Oh, that'd be great. I mean, I hope that the regulators and the companies are able to do studies that take that long to get it cured, because there's investors behind it and everything has to be quick, and which is unfortunate, actually. The second very short question is the kind of the blunted response in the REEF-2 study for patients being treated with the with the siRNA as compared to those with nukes alone. It's a very interesting study review because everything was stopped, really. Do you think that is a true effect or just a carryover effect of the siRNA, which has a longer half-life?
Yeah, it's a good point. So first off, I don't think the CAM did anything there. So I think it's all the siRNA, and I suspect we would see the same with other siRNAs. But I do think we need longer follow-up because we don't really know. I mean, it's been shown with other siRNAs that there's an off-treatment plateauing of response. So I think that's a real phenomenon, and we just need to understand what it is better to know whether it's durable. My suspicion is probably not, but.
Jordan, I have a question. You said that in natural history it was a slow decline in surface antigen, and that's not true of severe acute hepatitis or fulminant hepatitis, where the surface antigen plummets, and if you don't die, you recover.
Yep.
So perhaps new therapies are going to be as good as natural history if they don't kill you.
Yeah, I mean, fair enough, but you're starting with someone who's in a chronic state. So in that acute state, there's no question that is immune-driven clearance in acute infection. If you, I think when you're looking at chronic infection, it's just such a different phenomenon, but
you were talking about natural history of clearance of surface. Oh, you weren't talking about acute viral hepatitis.
No, I was talking about
you were
natural
talking
history of
about
spontaneous clearance of people that the sort of 1% per year of people who with chronic infection.
Number two.
Yeah, they're really different.
Sorry.
Yeah.
So Barbara Testoni from Lyon. So I also have a question for Jordan. So you just touched it very briefly. You said that inactive carriers have less responsive immune system. So do you think this is related to the time of exposure to HBS? And do you think this variable has to come in the game of new therapies? How can we relate to it? What is your thoughts?
Yeah, I don't know if they have less responsive immune systems. I think that those data suggesting that they're harder, at least in an ELISpot assay, to stimulate to get a good response from their T cells. I think that's a little bit hard to interpret. We've actually seen almost the opposite, that they're most responsive to PD-1 therapies, at least ex vivo. So I think it's a bit hard to say, but again, the other challenge is, what is the driver? Is it the fact that they've had longer infection, or is it the fact that they have low antigen levels, and that makes them that you need the antigen levels to actually maintain some degree of responsiveness? I must admit, I defer to the immunologists in the room, or you, to be honest, to answer the question.
So I have one question, really. There's a movement in the literature to treat more and more patients, anybody who's DNA positive. And Mark, you mentioned this group of young e-antigen positive patients. So is there a downside from an immunologic point of view to treating early? Do we need a little bit of antigen around to spontaneously to some of the outcomes that Jordan mentioned?
I guess I would argue they still have plenty of antigen around. But I think the question is, if you look at some of the data of long-term nuke therapy, when you start really early, and I have a few patients I've inherited from the pediatric hepatologist who have been on nukes actually since age 2 or 3. And what's remarkable for many of these, and this is obviously anecdotal, is just how low their surface antigen levels are after 10 years of nukes, started at a very early age. And it does make me, I wonder about what that would do for the number of hepatocytes infected. Because if you think about it, if you could reduce the burden of infected hepatocytes for curative therapy, it may help. So I do think there's an argument to be made for starting earlier with antivirals.
Not to mention that you decrease the risk of clonal expansion in cancer.
Yeah,
that is
right.
additional point.
Absolutely.
We can always keep it in the back of our mind.
100% agree.
Okay, Adam, you get the last question.
All right, good. So I'm going to come back to Mark on this because he made a comment before about the heterogeneity. We know the patients are very heterogeneous, but I would say that a lot of these combination trials now, they're sort of driving down S antigen DNA, and even in the follow-up period, they're getting to a lower set point. So do you think we can homogenize the patient population with these combination trials, and then after they're on follow-up, come in with sort of, so that way you're treating one patient population instead of a very heterogeneous population?
Yeah, purely speculative, but it's actually one of the reasons why I put kind of a lead in with siRNA or inhibit, to lower that set point before coming back in with either antivirals plus minus immune. It was a little bit what I was thinking about, but I can't point to a single shred of evidence to support that approach. But I was thinking about that idea of resetting patients with, RNA interference of some variety.
The only thing I would challenge on that a little bit is the question of whether it's really all S. Like, I think there is a focus that it's really all S, but I mean, as I sort of mentioned, you can bring down S in different ways, and I'm not sure doing it with a monoclonal antibody is the same as doing it with an siRNA, even though they're both targeting it, because the siRNA is probably targeting the other, well, it should be targeting the other antigens, and that probably is going to affect the way that they respond to it, whatever you're going to come in as your knockout punch to. So I don't know that you're homogenizing them.
I agree we're getting a bit myopic on HBS, but I mean, some of these follow-up studies, everything, the set points sort of reset lower, at least 6 months out from the follow-up, some of the stuff that I'm going to show later. But, so that's more the thinking. It's not, because it does all of it seems to be lower after these sort of aggressive combination therapies, even though they're not achieving cure.
Yeah, I think the point about not focus on S in terms of mechanism of action is important.
And with that, the last comment online was from Ashwin at Johns Hopkins, again, highlighting the importance of linking immunology with interventional trials so that we can find out in several sub-studies what happens on liver biopsy to things like cccDNA and integrated iDNA, surface antigen, et cetera. And so with that, I think the hardest part of Marian and my job was to control the vigorous discussion at the session. I think we were only 1 minute and 50 seconds behind, but please remember to complete the online survey, which will be sent to you later today, and there will be a coffee break outside in the foyer. Please return promptly at 10:20 a.m. Thank you very much.
There's no